Ladies and gentlemen, welcome back. And now I get to do something really fun. I have the privilege of introducing our keynote speaker today, Dr. Eric Olson. Eric is a member of the faculty at my institution, University of Texas Southwestern Medical Center. Eric, if I read your entire bio, there wouldn't be any time for you to speak here, so I'll just hit the high points. Eric was the founding chair of our Department of Molecular Biology. In addition to directing that department, he also heads two of our centers. Now, a center at UT Southwestern means that it has a staff of both scientists and physicians who bring their talents together to advance clinical science. He is the director of the Heyman Center for Regenerative Science and Medicine and the Wellstone Center for Muscular Dystrophy Research. He holds the Annie and Willie Nelson, yes, that Willie Nelson, professorship in stem cell research and the Robert A. Welch Distinguished Chair and the Pogue Distinguished Chair. Now, he and his trainees over the years have discovered many of the genes that control heart and muscle development and disease. His work, in addition to having been recognized in over 600 scientific articles, has also laid the foundation for what we call being a serial entrepreneur. He is a founder or co-founder of Miragen, Myogen, Lone Star Heart, and most recently, Exonics Therapeutics. Eric's contribution to over 600 scientific articles is in part why UT Southwestern was named by Nature as being the #5 institution in the entire world in its impact on industrial development. He is, and by the way, back to Willie Nelson. In his spare time, he plays the guitar and he leads a band called the Transactivators. And by the way, they are performing on October the 28th at the Herman Hall in Dallas. So without any further ado, I'm going to have Dr. Eric Olson come and speak to us about his work in Duchenne's muscular dystrophy. Thank you.
All right, I'm really happy to be here today. Thank you all for coming out, and I'm going to bring you up to date today on one recent area of exploration in my laboratory, which focuses on one of mankind's most insidious diseases, and that is Duchenne muscular dystrophy. I'm going to give you sort of a general high-level overview of our work in this area. So for those of you who are not familiar with Duchenne muscular dystrophy, abbreviated DMD, this is the most common fatal genetic disorder diagnosed in childhood. It affects 1 in 5,000 boys. There are 300,000 boys in the world afflicted with this disorder. It's typically identified by the pediatrician early in life, usually around age 2 or 3, when the mother will notice that the son is having difficulty on the playground or walking upstairs, and the physician will perform what's called the Gowers sign, which you can see shown here, where the boys have difficulty standing upright and have to use their arms to do so because of early degeneration of their muscles. And as you'll see, there is no cure for this disorder. This shows the clinical progression of this disorder. Once a mutation responsible for this disease has been identified in a young boy, there is no escaping the clinical consequences of that mutation, which progress as shown here with initial walking difficulties that then progress by the early teenage years to the loss of ambulation and confinement to a wheelchair. Ultimately, these boys have to be ventilated up to 20 or more hours per day, and ultimately this disease culminates in premature death, typically in the 20s from heart failure and respiratory collapse. So it's a really devastating disorder. The disorder is caused by mutations in the largest gene in the human genome, the gene that encodes the protein called dystrophin. This is a massive gene, 2.6 million bases. It takes almost an entire day to transcribe one single copy of the messenger RNA from this gene. And the gene, in part because of its large size, is subject to an enormous number of mutations. There are more than 3,000 mutations that have been identified in boys with Duchenne muscular dystrophy worldwide. And this is really one of the fundamental challenges in this disorder. How can you conceive of a genetic therapy, which I'll be talking to you about, that could efficiently and effectively address the thousands of different mutations that exist in this large gene? This disorder is inherited from mothers who are unknowing carriers of mutations in the dystrophin gene. You can see that schematized here. The gene is carried on the X chromosome. All women have 2 X chromosomes, whereas men have one X chromosome and a Y chromosome. So as you can see schematized here, this mother, shown with the red box on the X chromosome as a mutation in the dystrophin gene, she would be asymptomatic because she has a normal dystrophin gene to compensate for the mutation. But when this couple has children, 1/2 of the boys born to this couple will inherit the mutation on the dystrophin gene and will always develop the disease. As I said, there is no escaping the consequences of this mutation. 1/2 of the boys from this couple will not inherit the mutation, and girls from this couple, 1/2 would inherit the mutation, but because she would have another normal X, she would be a carrier. One of the really horrifying aspects of this disorder, which I hear about from patients frequently, is that because the disease is not diagnosed until age 3 to 4, typically, it's not uncommon for a couple to have had a second son before the first one is diagnosed, and that brings with it a lot of obvious serious issues for them to confront. The disease is caused, as I mentioned, by mutations in the dystrophin gene, and dystrophin functions as a molecular shock absorber for all the muscles in our bodies. Every skeletal muscle, which comprises 40% of your body mass, and the muscles of the heart, all require this protein to stabilize their membranes during each cycle of contraction. You can see that here in the lower right-hand panel. In green is a cross-section through a muscle tissue, and you can see that every muscle fiber has the dystrophin protein underlying the membrane where it's required to maintain a muscle function. In its absence, muscles eventually disintegrate, and this leads to the clinical manifestations that I described. So within the context of this talk, you can think about dystrophin as a molecular shock absorber that maintains the integrity of all the muscle tissues throughout life. There is only a single drug that has been approved by the U.S. Food and Drug Administration for the treatment of Duchenne muscular dystrophy, and that is a drug marketed by a company called Sarepta. The drug is called eteplirsen, and it received accelerated approval in 2016 based on the ability of this drug to increase the level of dystrophin protein in these boys from 0 to less than 1% of the normal level of dystrophin. So obviously this is a very minimal effect. This drug is currently sold to patients at an annual cost of approximately $1 million per year, and it requires constant infusion through an indwelling catheter in the chest of these boys and carries with it a number of side effects aside from the fact that the drug doesn't work very well. Nevertheless, this is the only drug approved for treatment of this disorder so far. Because my lab has worked on the biology of muscle development and muscle disease for over 30 years, we decided to ask whether we could take a new approach, not to treat the symptoms of this disorder, but to try to treat the cause of the disorder. How can we get at the underlying molecular mutations that cause Duchenne muscular dystrophy? And to do that, we decided to try to apply what I believe is the most revolutionary technology in decades in the field of molecular biology, and that is the technology of gene editing, which many of you or all of you have undoubtedly heard about, often referred to as CRISPR or CRISPR-Cas gene editing. This is a copy of the cover of Newsweek describing Genie and CRISPR editing. That article is a lot more interesting than the one at the top of this magazine, Trump's claim to China syndrome. So if you have not heard about CRISPR, you will hear about CRISPR. You can think about, for the non-aficionados in the audience, you can think about CRISPR as molecular scissors that can surgically identify and eliminate any mutation in the genome. Every cell in your body, as I think all of you are aware, contains 3 billion letters of the DNA code. And what CRISPR can do is it can identify even a single letter, one misspelled letter out of 3 billion. It can find it rapidly and it can correct that mutation. So this technology has obvious applications for a number of genetic diseases, and we're only a few years into this technology, but I think we can already see how it can be applied. And so what I'm going to talk to you today is about our initial efforts to apply this technology to cure muscular dystrophy. Without getting into all the details, I'd like to just summarize quickly how this technology works. This technology involves a 2-component system. On the left, you can see that there's a guide RNA. This is a short RNA sequence which one can direct to any sequence in the genome simply by identifying the complementary nucleotides to which you would like this guide RNA to home in on. The second component is a bacterial enzyme called Cas9, which can be directed to any sequence. It's extremely rapid, it's extremely efficient and accurate. It can be directed to any sequence in the genome by the guide RNA. And when Cas9 finds its target with the guide RNA, it will cut the double-stranded DNA helix, and then it will edit that cut in a number of ways that I won't get into all the details. Just as an aside, this system, the Cas9 enzyme in the guide RNA, is an ancient immune system that first evolved in bacteria as a way that bacteria could identify invading viruses and cut and destroy them. But molecular biologists have now been able to exploit this ancient immune system in ways to identify mutations in cells and even eventually in humans to correct monogenic disorders. So without getting into too many details, this is the structure of the dystrophin gene. The dystrophin gene contains 79 exons, exons being the segments of DNA that code for proteins. And these 79 exons are spliced together to ultimately give rise to the dystrophin protein, which is shown schematically at the top of this slide. And you can see the color coding of the different domains of the protein and how they're encoded by the various exons. If the shapes of the exons are fit together like a jigsaw puzzle, that tells you that the protein can be encoded across those different exons. These exons in red, there are 12 exons shown here in red, represent what are known as hotspots. These are regions of the genome that are especially sensitive to mutations that cause Duchenne muscular dystrophy. And these 12 exons account for about 80% of the worldwide population of boys with Duchenne. That's saying that if one could direct CRISPR to these exons, one could consolidate hundreds of the different types of mutations into singular or a few categories and could correct them by a relatively simple gene editing step with CRISPR. And so we've focused on these exons in our initial studies. This is the approach that we are taking at UT Southwestern. So a patient comes in, and I receive emails every day from mothers all over the world that tell me the mutations that their son has. Or if a patient comes in and has an interesting mutation and we want to know whether we can fix it by CRISPR, we would take a small blood sample from that boy and we can use a technique to convert that into pluripotent stem cells called induced pluripotent stem cells, or iPSCs, in a dish. And then we can introduce into those cells the guide RNA and Cas9, and we can ask, can we edit the mutation from this patient? And then with another simple step, we can convert those cells into muscle cells or cardiomyocytes in a dish. This is an extremely powerful technique because you can take the genome and a cell from an actual patient, and you can apply this technology to that actual mutation, and you can ask, can we fix the mutation? How efficiently can we fix the mutation? And if we do fix the mutation and produce dystrophin, is it correctly made? Is it stable? Does it go to the right place in the cell? And it also allows us to perform safety studies using cells from the actual patient that might ultimately be treated by this approach. So here's an example. This shows at the top exons 42, 3, 4, 5, and 6. This is one of the hot spots. In this case, we made cardiomyocytes from blood sample using that IPS method I described. This is from a normal brother of a patient. And we stained these cells for dystrophin in red, and you can see that all the cardiac muscle cells in this dish are expressing dystrophin. This is the brother. This brother has a deletion of one exon, exon 44, in the hotspot region. And when we made cardiac muscle cells using his blood to make stem cells, you can see they make no dystrophin. Then we make a guide RNA to skip the mutation. And then we look in the actual cells from this boy, the cardiomyocytes from this boy, and now you can see with one single editing step, you can restore the production of dystrophin by skipping over the mutation and putting the gene back in frame. And here you can see it at the bottom. This is a gel showing the dystrophin protein, which is massive. On the left lane, you can see the normal brother. There's the level of dystrophin that his cardiac muscle cells make. In the middle, you can see the affected boy with Duchenne muscular dystrophy. He makes no dystrophin at all. And on the right, you can see after treatment with gene editing, you can fully restore the production of this protein. So you can imagine the potential power of this approach. So this is my neighbor. His name is Ben Dupree. He lives in University Park in Dallas. Went to the same high school as my kids went to. That's his mother, and that's his cardiologist, Dr. Pradeep Mammon. Ben has a very interesting mutation. He has one single letter out of his entire 3 billion letters of his DNA that is wrong. And that letter is not even in the protein coding portion of the gene. It's in an intron between two exons. And you would think, one nucleotide in a non-coding region of the gene, what's the big deal? Well, that one error put him in a wheelchair. And he's become quite a celebrity in the Duchenne world. He's a very articulate spokesperson, has spoken at the FDA, and this is a poignant, title's a little bit confronting, but it's a poignant article written by the MIT Technology Magazine about our efforts to correct his mutation. So these are beating heart cells. So we've had Ben come into the lab. We took a small sample of his blood, and using that technology I described to you, we could convert it into iPS cells and make cardiomyocytes, and those are his actual cells. I can tell you, very few people in the world have ever seen their own heart cells beating in a dish. And so Ben came into the lab and saw his own heart cells beat, and that was a very meaningful moment. But not only that, we then used this simple gene editing technique to ask if we could fix his mutation. There's his cells when we created them from his blood. Those are his cardiomyocytes stained for dystrophin, and they make no dystrophin. And then here we treated them with a single cut CRISPR to eliminate his mutation, and it fully restores the production of his missing protein to normal levels in all these cells. And for Ben to be able to look through the microscope and see his own cells producing the protein that his body can't make was really a poignant moment. So this has generated a lot of interest. There's been many documentaries published worldwide about Ben and our efforts, and one of them, Dateline NBC, did a story that's quite a good one on this work with Keith Morrison. Some people think he's my long-lost twin brother. There with Ben Dupree. And we're looking at Ben's cells there on the computer screen. So this is the dystrophin gene again, and right here you can see exon 50 I've marked in black. This is the #1 hotspot for mutations in boys with Duchenne. 13% of all the worldwide population of the 300,000 boys with Duchenne have mutations in exon 50, and that's exactly the same exon that the Sarepta drug, eteplirsen, was directed against to try to skip the mutation and put the protein back in frame. There was no animal model to study this common mutation, and so we decided to create one and then ask if we could optimize gene editing in an animal to fix this type of mutation. So we made a mouse with we call it a humanized mouse. It has this exon 50 deletion. Again, on the left, you can see dystrophin protein in green in normal muscle, and on the right, you can see this mouse where we've created this human mutation, and it makes no dystrophin. So this provided us with a very powerful platform for testing really the effectiveness of gene editing in a living animal. And so to do that, we had the challenge: how could we deliver the gene editing components to a living animal, in this case a mouse, but we hope eventually that might be a human. And we were enabled in this effort by the existence of a virus called adeno-associated virus. This is a harmless virus. It's been approved for use in people by the FDA, and it's been in thousands of people. It's completely harmless. And there's a form of AAV called AAV9, which fortuitously infects muscle and the heart specifically, but not other tissues in the body. And so we engineered into AAV9 the CRISPR gene editing components, the guide RNA and Cas9. And then we asked, if we introduce this into mice, in this case, by injecting it, could the virus, shown here in green, selectively find the affected muscle cells in the body, deliver their cargo to those cells, and could these molecular scissors go to the nucleus, find the DNA sequence that's mutated, and fix it? And I'm going to show you the results of those experiments. So this at the top shows 3 representative muscle tissues from a normal mouse. The triceps, that's a muscle in the arm. The diaphragm is a really important muscle when you think about muscular dystrophy, because these boys ultimately die from respiratory failure due to diaphragmatic destruction. And you can see the heart. And in the middle, the lower 3 panels, you can see the muscles from this humanized mouse with the most common human mutation, and they're completely devoid of dystrophin. So this represents a very powerful model. So we injected these animals with the virus encoding the CRISPR contents, and we looked 4 weeks later after systemic delivery by intravenous or other types of delivery. And here's the result. You can see that this gene editing approach can essentially fully restore the production of dystrophin. It's efficient. It affects all the muscle tissues, including the heart of the body, and it's rapid. This is 4 weeks, I believe, after the initial injection. This shows quantification of the level of dystrophin protein. On the left are 2 normal, this is from heart, 2 normal mice. That's the level of the dystrophin protein that's very large at 250 kilodaltons. In the middle 2 lanes are the mutant mouse that has the humanized mutation. There's not a whiff of dystrophin in those animals. And then on the right shows the level of dystrophin protein that's restored within 4 weeks after delivery of the virus systemically. And this is sufficient to bring back about 80% of the normal level of dystrophin. It's been estimated that in boys with Duchenne muscular dystrophy, if one could restore 15%, 15% of the normal level of dystrophin, that would be curative for this disorder. It's not going to be necessary to bring back 100% of the dystrophin protein. Even a relatively modest rescue could have profound therapeutic benefits. So what are the distinguishing features of this approach relative to all other therapies that have been attempted and have failed in this disorder? One is that CRISPR-Cas9 gene editing corrects the cause of the mutation. Once the mutation that causes the disease is potentially removed, then the cause of the disease is eliminated. We believe it represents potentially a one-time treatment, particularly for the heart, because the heart cells that you are born with are the ones that you live with for your entire life. And this method works especially efficiently in correction of the mutation in heart cells, so we believe that this could be a one-time treatment. It potentially reaches all the affected cell types. I've shown you using AAV9 as a delivery vehicle for muscle and heart that we can essentially permeate the entire musculature, at least, of a mouse. And based on optimized guide RNAs that we've now generated, we believe that there's the potential to correct up to 80% of the mutations responsible for this disorder in boys worldwide. There will be a small subset that cannot be corrected by this approach. If, for example, a boy has a very large genetic deletion that eliminates essential domains of the protein, it can't be fixed by this approach, but fortunately that represents a relatively small subset. So this is where we are right now. I've shown you evidence that we can correct human mutations in actual cells from patients, and we can demonstrate the ability to restore dystrophin from those patients in a dish. I've shown you that we can create now mouse models of Duchenne. I gave you one example, but we now have a collection of mice with the top 5 most common mutations that represent roughly 40% of the worldwide population. We can efficiently correct those by a simple one-time delivery of gene editing components using adeno-associated virus 9. The next challenge is to scale this up as a prelude to potential eventual treatment in humans. To do that, we've scaled this up and have tested whether we can correct Duchenne mutations in a dog model that harbors the exact same mutation as the most common one I showed you. And I won't show you that data today, but I can tell you that it's remarkably efficient. Within 6 weeks after a single injection, we've been able to restore dystrophin in the injected muscles to roughly at least 80% of normal. So we're optimistic about that, although there's obviously still much work to be done to ensure safety of this approach. So the Duchenne community is really a warm and wonderful community, which my laboratory members, some of whom are sitting here, and I have gotten heavily involved with. And this is at a recent meeting of many parents and families supported by the patient advocacy group Cure Duchenne, where we talked to them about our work. And I can tell you that the excitement in the Duchenne world about this new technology is palpable, and there is still much work to be done, but I think based on the work we've done so far, we're really optimistic. This is a real picture. Recently, this is looking up in the sky above Dallas. This was a cloud in the sky above Dallas. Looks to be a double-stranded DNA helix with a double-stranded break. And there's actually where it was. So I sent this out on Twitter. It went viral on Twitter. The only thing I ever sent out on Twitter that went viral. Spontaneous DNA cloud with double-strand break above Dallas, a good omen for CRISPR gene editing. And that's not a photoshopped picture. It was actually taken by Yi Li, who's sitting right there in the audience. So this is my lab here on the right, and we've been supported, I should have mentioned, we've been supported by the Wellstone Muscular Dystrophy Clinical Research Center, which is funded by the National Institutes of Health at Southwestern. We've been funded by the Center for Regenerative Science and Medicine. And lastly, in order to move this work forward towards eventual clinical application, we started a new biotechnology company called Exonics Therapeutics, which was initially funded by a patient advocacy group. And I really want to publicly thank Frank Grasler. He's given me a lot of great advice and support along the way in launching this company, and we are really excited about the potential to take this technology forward as an eventual treatment for the many boys and families that could benefit from it. And lastly, all this work was done at UT Southwestern, and happy to take any questions. Thank you.
I'll start with a question, if I may. Eric, is it too early to forecast or predict whether what you were learning through this science can be applied to any other genetically based diseases?
Yeah, so Frank wants to know just what is the long-term potential to correct monogenic disorders by this approach. I think the potential is enormous, and there are many groups and companies that are using this technology to treat monogenic disorders such as sickle cell anemia or cancers or other things. We feel that Duchenne represents an optimal situation for testing this technology for a number of reasons. One is because, without getting into too many details, one is because the central region of the shock absorber of the dystrophin protein can tolerate many types of deletions. So you can excise mutant regions of the gene and put it back together very simply and effectively, as I showed you. Secondly, this is a disorder, as I described, that for which there is no escape. And as soon as the diagnosis is made, the parents know what the clinical progression will be. And I think disorders like this really lend themselves to these sort of, this is about as cutting edge technology and probably about as profound a technology as you can imagine to actually change the human genome. So I think this lends itself especially well to that. And lastly, because the tools exist, as I showed you, to deliver these gene editing components with high specificity to the affected tissues, I think it's a really optimal setting for testing that. But there are a lot of safety studies and other things that we need to address before clinical translation. Yes, Melissa.
So, two questions. What's your best guess at this point about how soon you'll be in phase 1? And secondly, what are your thoughts about, over the long term, what parts, if any, of the company that you formed you would expect to remain in North Texas?
Those are both difficult questions.
What do you expect?
It's a loaded audience.
How long to get this into phase 1? We do have a timeline on that. We've engaged regulatory consultants, and we're completing long-term safety studies in dogs right now, as well as in mice. So far, we've not seen any evidence whatsoever of any toxicity of this approach. We have not seen an immune response, and we've done extensive deep genomic sequencing to ask, are there unexpected so-called off-target effects in which Cas9 might cut the wrong place? We've not seen any off-target effects in human cells, in mice, or in dogs, and neither has anyone else that's been reported. So far it's safe. I think the challenge to move it into humans will be to further ensure safety, but also then to scale up. And I think it's a good omen that we could scale up from a mouse to a dog and maintain the high efficiency. The next step is a big one. I believe that within 2 years we can be ready to launch a clinical trial. But you can't hold me to that because there's obviously a lot of uncertainties along the way. And patient selection of an eventual clinical trial, if this gets there, will be key. Ultimately, if this type of technology were to work, the earlier the intervention, the better. Once the muscle is gone and there's nothing left to fix, it's a tough situation. So what I would hope, my greatest dream, would be that if we could demonstrate efficacy and that immediately upon diagnosis that this type of technology could be brought to bear on this type of disease. But that's obviously a long way in the future. Your second question was, what's the likelihood that this company will be placed in Texas? Right now, Exonics Therapeutics is a virtual company. It's being run essentially by scientists. It's based on science in my laboratory that's being leveraged to start the company with regulatory consultants. But I think the ultimate location of the company may not be in Dallas. It may be elsewhere, and we'll have to see about that. Other questions? Okay, thank you very much.